Πολύ ωραία. Για την παρουσίαση του επόμενου μιλητή θα μιλήσουμε στα αγγλικά. It is our great pleasure to welcome Maurizio Dattilo to today's annual scientific meeting. Mr. Dattilo is a prominent Italian researcher and now the CEO of Parthenogen SAGL, and we will do our absolute best to follow your chain of thought and presentation on a very cutting-edge topic on the importance of vitamin B6 and gasotransmitters in the functional aspects of the endometrium.
Thank you, Mr. Chairman. Ladies and gentlemen, good morning, good evening, kalispera. My topic of today is very new for you. I will try to be didascalic and to keep it simple. Let me start immediately. I cannot consume time. We are late. And so I'm going to speak to you about vitamin B6. Vitamin B6 is one of the main essential micronutrients. We have to eat some every day because it is essential for the function. However, this is not a critical micronutrient. I must say that in Western countries, an absolute shortage is really rare and difficult if we are normally eating. Just to know what we are speaking about, how does it work? Very simple and fast, because we do not really understand. Vitamin B6 is used when an electron has to pass from one substance, one molecule to another. It is a temporary receptor, give it back, and the job is done. It is called shift base. It is not consumed. It remains there. So then we have the problem when the activity is too much. How do we stop it? This is part of my topic later on. However, no vitamin B6, no life, no way. Why? Because this is involved. This is a very restricted list, of course. Don't worry, I'm not going to speak to you about all of that. Today I can focus on two aspects that are the same. That is the metabolism of amino acids. So all of the proteic metabolism, just to recall you, transaminases that you usually test in your patients, they use B6 to transform one amino acid to another. And in this process, one of the side products that is actually the main product we know today is the release of gasotransmitters. And this is a word that is new to many of you. Gasotransmitters are small gaseous molecules. They are ubiquitous in the metabolism. They are released every tissue, every cell, continuously. What is the particular? They are a gas. They permeate, diffuse everywhere, every compartment, every cell. They have no limits, no transporter. They go everywhere. And what they do, they modify the activity of proteins. It is a huge effect that we did not see. There is a puff of gas, and suddenly thousands and thousands of proteins change their function, sometimes the opposite. It is an entirely new discovery in metabolism, and indeed, this resulted in the Nobel Prize in 1998 for the first discovery of nitric oxide signaling in cardiovascular disease. Since then, it exploded. This was now in Medline, titled abstract. You find 220,000 papers published. Well, you should know that 1 out of 3 is about reproductive physiology and pathology, and we don't know yet all of this. This is still out of our knowledge. We didn't see them because they are vanishing. They are gases. They last seconds, maximum minutes. So that's why we did not see them for decades of research. You see the half-life of the main. These are the 3 main ones. We are discovering a few more. All of them interact with the metal centers of proteins. So all the metal proteins are the targets, starting from hemoglobin, the guanylate cyclase, the most famous enzymes are managed by these gases. Hydrogen sulfide has a wider action because it makes a persulfidation, a small modification of free systems of the proteins, and they change their activity. I'm going to show you something. And as I told you, this is extremely relevant for human reproduction. You're lucky, I'm not going to go through all of that. This is a review from a few years ago, but it is still a good guidance to start understanding what is happening throughout the reproduction with these gases. And the release, back to vitamin B6, the release of this gas, H2S, for you to understand, this is the smell of rotten eggs. That is hydrogen sulfide. It was known as a poison, killing people in caves, things like that. Now we know it is a biologic gas. It is produced, this is very important and very rare in biology, dose-dependently, strictly dose-dependently on the amount of active B6. That means that is a wide variety of response. There are enzymatic release, and I think the most important is the non-enzymatic, kind of, because it is the one in equilibrium with diet and with iron metabolism. And these are, the non-enzymatic occurs into the vessels, and the most important one for the endothelial dysfunction. Speaking of pregnancy, again, this is too wide, not my topic today. You should know that hydrogen sulfide triggers all of the main changes that are needed for a pregnancy to occur, from the follicular rupture to the induction of endometrial quiescence, from the ovulation till the labor, the whole time, immune tolerance, angiogenesis, and vasodilation in the utero-placental district. All of this is managed by this. Today, I am focusing on the inflammation part, on the immune tolerance, to be more restrictive. Immune tolerance is the basis for reproduction. The sperm is allogenic, should be immediately destroyed. The embryo is semi-allogenic. Again, it should not be tolerated, and this is a special issue in oocyte donation, where the embryo is entirely allogenic, and the placenta are entirely allogenic. And here, the immune challenge is the main one. The eclampsia risk in oocyte donation is 8 times. It is the strongest risk. This is possibly the reason. Immune tolerance is managed by the differentiation of T lymphocytes. This is the main narrative against it. We know that it is epigenetically regulated, and there is a specific gene activation that is gene tolerant. It is called the induction of Treg lymphocyte, T regulatory lymphocytes. To simplify, these are those controlling autoaggression. Very simply, the differentiation to Treg is marked by the activation of a gene, the FOXP3 gene. We can follow it by the gene or with the protein, the FOXP3 protein. It is activated, and the lymphocyte becomes a T regulatory lymphocyte, to simplify. Very contemporary topic. This is the reason for the few weeks ago for the release of the Nobel Prize in Medicine of this year for the discovery of Tregs. This is really the hot topic of the moment. And the activation of the FOXP3 gene is epigenetically regulated by hydrogen sulfide. It is the activator. There is a puff of gas in a tissue, and the lymphocytes around activate FOXP3 and becomes T regulatory. This is an extreme simplification, but we are among clinicians. We have to understand the picture. Breaking news, I was just discussing this with Michael Giannakis. We have now discovered that we also have B regulatory cells. They also differentiate in regulatory plasma cells, releasing antibodies that are intended to induce immune tolerance, still under investigation. There is no single gene here to track, it is much more complex, but also this differentiation is triggered by the release into the tissue of hydrogen sulfide. And you understand this must be, speaking of reproduction, this must be districtual. We need to have immune tolerance within the uterine environment, not everywhere into the body. It goes also everywhere. We know that during pregnancy, several autoimmune diseases can go in regression just because of this activation that is in part also systemic. But beyond Tregs and Bregs, that is immune cell-mediated inflammation, we know of a wide actions of hydrogen sulfide in the inflammation. You see the title of the paper here, Mediator of Resolution of Inflammation and Injury. How is it possible? It is possible because, as I told you, the persulfidation, the modification of the proteins, it is relevant especially for membrane carriers and pumps. The most studied one is the K-ATP channel. This is the pivotal paper from almost 20 years ago. The K-ATP channels are ubiquitous. They always do the same job. Polarization of the membranes, especially in muscular cells and in neurons, of course. To make the story simple, when the gas binds one of the subunits of the pump, the K-ATP is the blue one, the channel remains open. When the channel is open, the membrane is hyperpolarized, there is no influx of calcium and no transmission of the impulse. Speaking of leukocytes, when the channel is closed, no calcium influx, they remain fluctuating into the blood. When the channel closes, they attach to the endothelium and they start releasing inflammatory cytokines, chronic inflammation, and endothelial dysfunction. Okay, so this is a key aspect of inflammation. Other membrane pumps and channels are also involved, and keeping it as simple as possible, but this is by distance the most important mechanism. So the gas activates the immune cells to differentiate to the regulatory tolerant type, works on the K-ATP channels to block the release of the inflammatory cytokines. The reverse is also true. You remember I told you this is not consumed. How do we stop it when it is too much? Well, inflammation reduces the concentration and the activity of vitamin B6. We know this very well. There are many papers showing it, even if it was difficult to understand. Now, you have to understand that vitamin B6 has only one active form. We call it P5P, pyridoxal 5-phosphate. And you see that the circulating level of P5P, of active B6, strictly correlates with the inflammation index. Do I have a pointer here? Yes, no? Oh, okay. You see that whatever the intake from feeding, the shape is the same. They go to decline. Of course, if you are starting with a better dietary feed, you end up with a higher level, but the destiny is the same. As inflammation increases, it goes down. How does it work? With the diet, we eat only inactive vitamins. Pyridoxine, pyridoxamine, pyridoxal. These inactive vitamins are activated into our cell, into our body. There are specific enzymes doing it. By the way, these enzymes circulate with the defective variants, very common. This is another side of investigation. And they produce active B6. On the other side, inflammation activates enzymes that deactivate the vitamin. And this is fundamental. Otherwise, we should not establish phlogosis districtually when it is needed. And why it doesn't go? Doesn't progress? What happens? Okay. And it is alkaline phosphatase. And I also want to recall, you see alkaline phosphatase in every lab report when you send your patients to the lab. It is a mystery. We know only 3 substrates for alkaline phosphatase. Phosphatidylethanolamine, it is the synthesis of fatty acids, to make it simple. Inorganic pyrophosphate, we suppose it is involved in the providing phosphates for bone mineralization. And then pyridoxal 5-phosphate. It is the only job of the vitamin. It is of the enzyme. It is called alkaline because it works at alkaline pH. And actually, the optimum pH is 8-10. So, first, mysterious. We don't know the exact role in bone metabolism. We don't know why it increases in cholestasis. Simply, we don't know. It is a finding. And the normality values in your lab are established to detect cholestasis and bone activation, not to detect inflammation. This is important to tell you. We will be speaking within the normal limits what happens. So optimum pH 8-10, but our pH is 7-4. Why? Where does it find it? In mitochondrial matrix only, and in areas of inflammation. The enzyme is increasingly released when there is inflammation, and here it will be working. There will not be dephosphorylation elsewhere, apart in the area of inflammation. So, to make the story simple, diet, we get pyridoxine here, phosphorylation, active form, dephosphorylation by alkaline phosphatase, we revert back to the inactive form. Now, an interpretation, this is speculative, we cannot prove all of it, but makes a lot of sense. You have a reason for inflammation, it starts, ALP is released, dephosphorylate B6, this helps less of release of the gas, as I was showing you, that is the anti-inflammatory one. At that point, you can have a recovery. It's balanced. If you are young, you have a good diet, and your genetics is not so terrible with the enzyme, you heal. But if you are on the other side, what can happen? You have a low B6 reserve, your diet was not the best, the individual genetics is not the best, maybe advanced age, you are weaker in this response. What happens? That there is a little inflammation does not go under control, it increases, and then you have a vicious cycle, which is the chronic inflammation. This is one of the interpretations of chronic inflammation. How does it develop? It is speculative, no final proofs, but it makes a lot of sense. And if you look at pregnancy, you see that the physiologic dynamics, we know of the underlying low-level burning inflammation of pregnancy as a physiologic aspect of pregnancy. You see that throughout the pregnancy, alkaline phosphatase goes up. In parallel, active B6 goes down. After delivery, everything goes back to the normal. But this can take a pathologic derangement. We know something. For instance, we can look at what happens in hypertensive disorders of pregnancy, and you see that the level of alkaline phosphatase within the normal limits, please, because that were for cholestasis, forget it, tell us nothing. Within the normal limits, it is a sharp increase compared to normotensive, and it is almost entirely due to the increase of placental isoenzyme of PLP that indeed has 100% sensitivity in detecting the disorder. They are strictly linked. And we also have some evidence. This is a study in bovines where you see that there is an endometritis in bovines. They treat with antibiotics, as always, and you see that BT is before treatment, AT is after treatment. After treatment, the pH from alkaline goes back to neutral, and alkaline phosphatase from high level goes back to normal level. This is the healing of inflammation. We have very direct evidences on this. And to go back to the gas, no, wait, this is extremely relevant to the whole pathology, to the whole reproductive pathology. This is a beautiful review from the Spanish colleagues. I invite you to look at that. Specific disease, you see on left and right, and then practically all the problems that we are facing are related to this inflammatory status, starting from implantation failure, of course. It is the most evident one. Back to the gas, experimental models are telling us that all of this is reverted when the animals are injected a chemical donor of the gas, of hydrogen sulfide. You see, for instance, I put larger the endometrial thickness. There was an injury causing the inflammation. This causes a thin endometrium. They administer the gas. It goes back to baseline. So, seems to be the mechanism. Next question is, how do we activate it? Of course, yes, we eat vitamin B6 with the food, but the real problem is whether we balance the activation in the right way. So a good diet is important, but it is not usually the cause of the problem. Supplements, wait a minute. Drugs, do we have drugs for this? This is the dream of the drug industry. Practically every major has a project, usually outsourced in secondary branches because they are afraid of failures that causes the stock to fall down. Just to give you an idea what they are looking at natural donors, garlic. Do you remember the singers at the opera that eat garlic? Because it is bronchodilating because of the gas of the hydrogen sulfide and onion and so on. But we cannot produce supplements or drugs because they generate byproducts that are toxic. Then there are the chemical donors that we are using into the experiments, in vitro and in vivo, but they're not suitable for human use because it is one-shot release, and this can be toxic if it is too much. And then they are developing drugs. The problem is that it should be at the right place at the right time only, and it is extremely difficult. They are trying a series of, but I can tell you in the next 10 years we will not see anything from the drug industry. And this is just to show you what they are doing. This is an example. In this case, they made a liposomal delivery of a chemical donors that should go straight to the colon. To give you the idea how complex are these developments, not our topics, just to show you. But we are doing it with micronutrients. This is my patent, just to disclose it. You see, I also claim it for repeated implantation failure and miscarriage and infertility. Usually they do not approve these claims, but it is there. How does it work? How can we do it? It is stupid simple when you know it. Cysteine is the main substrate, the SH group of cysteine in the catabolism of the amino acid to release the gas. It is dose dependent on the amount of activated vitamin B6. We learned this now. I also included taurine, small amounts, because, and this is the key of the story, because with small amounts of taurine, I can give a negative feedback to the enzyme there. It is cysteine deoxygenase. When it is activated, it is induced at 40 pair on baseline. If I give a little taurine, I stop it down and allow cysteine to go to my target. So this is, to be honest, a manipulation, because I'm using a supraphysiological level of activated B6, and this inhibitory effect of another substrate. Does it work? Yes. I published the validation. We had to do it in mass spectrometry, usual model. Subjects were sampled. We made the whole metabolome by mass spectrometry, 10 days of treatment, and then again the metabolome to see from the reporters, metabolic reports, what was activated. And we found a huge activation with this. Also, although it was not for everything, to show you, I told you there are enzymatic pathways here on your left, and they are activated in response to specific issues. The CBS pathway responds to protein misfolding. It is part of the unfolded protein response, and possibly to regulate the redox balance. The other enzyme regulates homocysteine postprandial, and it uses homocysteine to release H2S. We modulated the other two. The first one is the mitochondrial pathway. I did not tell you this, but H2S is a substrate on the respiratory chain instead of NADH to feed the proton gradient and to produce ATP without release of reactive oxygen species. This is the clean energy of our cells. And then we activated strongly the non-enzymatic release intravascular. That is the one in equilibrium with diet. This was expected, but the activation was massive. This is now available in a marketed product from the sponsor of my session. And this is the product that I patented. Here is I told you already of the composition, cysteine, taurine, and pyridoxal 5-phosphate. The dose that we are advising is, let me go there. Sorry, this is extremely important. Please look at this every time you use a supplement. GMP manufactured, in-process control, release control, stability studies. Otherwise, use a supplement, doesn't work. You never know if your idea was wrong or the supplement was fake. Always ask this to the manufacturers. This is extremely relevant. See this, 2 tabs per day. In the study I showed you, we were using 4 tabs per day. I'm currently taking 3 tabs per day to control my chronic inflammation. Should be taken between meals because they are hydrosoluble substances, but in some cases, I'm using it in some patients postprandial because it gives better control of homocysteine, vegan friendly, no allergens, no gluten, no lactose. To do what? Well, I told you, this impacts. Look, for instance, this very well-written review. They released 2 reviews, one for males and one for females. It impacts almost everything. The question is, you are physicians, you should know who is the patient. Well, here we have some path to go. Of course, the first thing you will see, I will look at vitamin B6 level in blood. This is a problem. First, it is offered by labs. It is a little expensive. But the common test does not distinguish between activated and inactive vitamins. So since the problem is there, does not inform you. And as I told you, it is very rare to find an absolute shortage in our countries, in our patients. And then there is another problem, that the shortage, I showed you some papers, only the titles, is usually districtual. I can have a deep shortage in the testes. I see nothing into the blood stream because inflammation is there. Okay, so this is the problem when we deal with this approach. You can look at the markers of chronic inflammation, as I told you, high sensitivity C-reactive protein, VES, and the cytokines. If you use, as we do sometimes with women with implantation failure, if you look at that, you will find signs what is happening there. The inflammatory cytokines are increased, the anti-inflammatory cytokines are depressed. There is a new index that has been created in oncology. It is called the albumin to alkaline phosphatase ratio. they discovered that when it is the ratio of the two, so the lower the albumin and the higher the alkaline phosphatase, the higher the inflammation background. There are more than 100 papers published in oncology. This correlates with the prognosis, to the response of the treatment, to the relapses of cancer. It is the chronic inflammation. You can look at metabolic markers. I have no time today to tell you all of the details on these. Of course, homocysteine is the main marker. Increased ferritin, because the intravascular release manages in some parts the iron metabolism, okay, and then you can have accumulation of ferritin if it doesn't work. D-dimers, coagulation, and of course the absolute level of alkaline phosphatase. If you find it at the upper level, you start being suspicious. What I've been, of course, I have only the clinical study for the validation. Then we are now running experiences in a series of situations. In a couple of cases, there are studies starting. In luteal supplementation, if you have ladies with that type, the schedule is pretty simple, just like you do with progesterone, because, you know, in implantation failure, in most cases, they have progesterone. We add more trying to force a blockade, but the blockade is very likely into this secondary effector, and if you increase it, you can address the problem. In the teen endometrium, but we don't have yet clinical data on this, only experimental data, and in this case, we should treat continuously for several cycles. In every endometritis, there are some experiences ongoing even here in Greece on chronic endometritis. We are getting surprising results in PCOS. And of course, I have to specify those because PCOS is an ocean of diseases. I'm speaking of those with true insulin resistance and hyperandrogenism, and especially with high LH. Whatever you do, you can never revert down LH in these ladies. We are seeing very high LH going below, within the normal parameters, and all of it disappointing. And very likely it is because we antagonize the follicular arrest. They are able to progress, and this improves everything. And finally, the hypertensive disorder of pregnancy. But this is obstetrics, not our job, usually. To conclude, hydrogen sulfide is a physiologic gasotransmitter that regulates pivotal homeostatic functions. By the way, those are much relevance to our practice, immunity and inflammation. Its release is dose-dependent of the availability of activated vitamin B6. This is indeed extensively inactivated in areas of inflammation. This is one of the mechanisms of chronic inflammation. Chronic inflammation is involved almost in every reproductive problem, but we can activate this release now with specific micronutrients. I showed you. That is the brand of our sponsor. This is, I wrote this yesterday when I was coming here. I really think this. I am surprised because I've been working with dietary manipulations for decades, and whatever we do, it is compensated. So it is a little frustrating because metabolism accommodates everything, and every road brings to Rome. This is metabolism. For the first time, we are really modulating fundamental homeostatic functions by a dietary intervention. I'm excited. We have new avenues of research. Who wants to collaborate? Please contact me. I will be happy. We are triggering a lot of this. Thank you for your attention.
Thank you, Mr. Maurizio. It was a really very insightful and interesting presentation. Of course, like clinicians, we are not able to fully understand all the aspects of cellular metabolism, but one question that I would like to address to you is related to liver detox, liver enzymes in relation to increased ALP, and also the enhancement of mitochondrial energy. If these two things, like a combination with your medication, for example, can make a really big difference. Thank you.
Okay, first of all, the liver. I am a hepatologist, but we don't know. We only know that the arrangements of these lead to steatosis, because the synthesis and release, especially of lipids, is affected, of triglycerides is affected. That's all. I have no other answer. Mitochondria is a fundamental aspect. I told you, our cells have the hybrid engine. We use glucose, fatty acids as the massive energy, but we pay a bill of reactive oxygen species. In some conditions, mitochondria shift to another energy source, that is this one. I give you an example. For instance, metformin. We never knew the mechanism of action. Well, metformin blocks the section 1 of the respiratory chain. Blocking it, it triggers the alternative metabolism, that is this one. It triggers hydrogen sulfide. Of course, it is not enough. That's why we do not solve anything. Many other drugs that we consider antioxidants, they are not antioxidants. They block the respiratory chain, and this is what happens. What is the physiologic meaning of this? It is possibly to counteract reductive stress. My next speech. Too long.
Thank you very much. Any questions from the audience? Excellent. Thank you very much, Mr. Dattilo.